Hello students, how are you today? How are you doing? Are you good?
Eat KFC.
Eat KFC. Oh, nice, nice. I also want some KFC. But let us, before we go into some of the nicer things, let's quickly go through our science. Today is science, and who can tell me what have we been doing in science? What have we been doing? Who wants to guess or who knows? What have we been doing? Okay, I'll start off with revision quickly of the previous lesson. Remember, we did the types of propagation. I want you guys
Teacher,
to tell me.
and what page?
That's the interesting part. You have to go look for yourself. It should be on between page 60 and 64. Between page 60 and 64. 6 type of propagation. 6 type of propagation.
Between 60 and 64.
Yes, somewhere there it should be. But we can play a bit of hangman and you can guess as well. Let's play a bit of hangman. Anyone, give me a letter. Give me one letter. Anyone.
What?
Give me one letter.
Teacher, between 60 and 64.
Yes. Today we will start on page 65. But someone give me a letter.
A.
A. A.
Yes. Four.
Give me another letter.
S.
S.
S. E.
E.
D. D.
D.
Sitting.
Seeding.
Sitting.
Seeding is the first form of propagation we looked at. The first type of propagation we looked at. What is number 2? Let's start.
Cutting.
Cutting. Okay, so we have it now. Let's complete them here. Oi. Go back. Insert text box. Why don't I insert my text box? Stop share quickly. Go back here. First one we said was seeding.
Sitting. E.
Bigger.
ไม่ใช่ cutting C U T Sitting. Sitting.
I N G.
G.
What is the second one?
Cutting. Cutting.
Cutting.
Cutting.
Cutting. Yes.
Copy. T I N G S.
Cuttings. So let me just change this. Oh.
Layering. Oh, ทำไมมันคัตติ้งล่ะ layering copy อ่ะอะไรไม่ได้ก๊อป
Layering. Yes. Next one.
build budding ก็จะพูดคำว่า building และ budding budding,
Budding. Next one.
cooking, plugging,
Cooking.
plugging,
Plugging. And the last one.
Enriching,
Enarching. Yes, that was what we looked at when we looked at propagation, types of propagation. Now let's look at some technology that people use in propagation, especially to help them make more plants faster. This is also very helpful for farmers because you can create more plants faster and grow more fruits and vegetables and sell them. Let's look at some advantages of propagation. Number one, get new species of quality plants. Yes. Number two, conserve endangered species because you can just breed the endangered species and keep their genetics unique. Number three, increase the number of plants. We want more plants, more oxygen, longer we can live. Number four, help improve the plant species. With propagation, it can help to get rid of some weaknesses in plants. So some plants might have a weakness to certain type of insects, and it might make that plant less less susceptible to insects. So it will live longer without any problems. So 2.1.5 on page 65, technology of propagation. Let's have a look at what they say. Currently, there are many uses of technology, especially biotechnology, to help propagate and improve plant species to increase their products and improve the quality of plants. So biotechnology is using living creatures or a part of a living creature, such as a plant, animals, and microorganisms, and applying knowledge of scientific methods to produce other products that are useful for humans. Biotechnology for propagation and species improvement include tissue culture and genetic modification. Now, this is a lot of big words and a mouthful of information. Let me explain it to you a little bit easier. So biotechnology is just where we use science to help improve plant production so we can have more plants and help the genetics and the type of plants to become stronger to different environmental issues. So this is biotechnology. We use it to create more plants faster so we can eat more food. And we also use it to make the plants more resistant to nature so that they can live longer and healthier than they used to live in the same conditions. Okay, now tissue culture. Where do you think, in which type of propagation would you find tissue culture? Which one of the 6? There might be more than one. Who knows? Who wants to guess? One plant of tissue, one form of tissue culture can also be cuttings. And we'll watch a video soon, then I'll show you the cuttings. Okay, so tissue culture uses tissues from growing parts of a plant, such as stem tips and root tips. This is then grown in a carefully controlled manner. This kind of propagation can help increase the number of plants in a short period of time, and it can help us get plants with the same characteristics, such as orchids. Our little friend in this pink blocks here, in this pink voice bubble, what does he wrote down here for us? Cultured tissues are divided into groups of cells called calyces, which can develop into plants. Now we'll have a look at a video in a bit after the next genetic modifications. Then we can see a little bit how they use these type of techniques. Now, genetic modification involves cutting out the genes from one organism and putting them in a different organism. This helps produce new organisms with the characteristics we want. That's if you know a little bit about chilies. You get chilies that are not spicy at all. You get some which are a little bit spicy. You get the bird's eye chili, which is quite spicy, the Thai chili. But then you get super spicy chilies, like the Carolina Reaper or the ghost pepper are super spicy chilies. And those chilies are made through genetic modification. By using crossbreeding hot chilies to find genetics that they want. So they want to make it spicier and spicier. Okay. The last one we're going to look at now is cloning, and then we'll watch a video. Cloning is another method of propagation. We can clone a plant by cuttings. Cuttings taken from a plant and grown will have exactly the same genetic characteristics as the parent plant. So you make an exact copy. We can see that by using biotechnology for plant breeding, we can purposefully select the characteristics we want in the plants we wish to grow. It also allows us to easily take good characteristics from one organism and combine these characteristics into a plant. A little voice bubble at the bottom. There are many ways in which we can selectively breed or propagate plants, or even genetically modify them in order to produce new plants that have the characteristics we are interested in, such as resistance to disease. Yes, that's what I said. Resistance to disease. Let's quickly watch a video about cloning. A short video which will show us about cloning. Okay, let me just... Modern cloning techniques. Let's make sure there's sound. Share sound, okay.
Can you spot the two clones? When we talk about clones in science, we mean organisms that are identical copies. They have the same DNA as each other. Identical twins are examples of naturally occurring clones. Both plants and animals can be cloned. Let's start with plants. A gardener, farmer, or plant breeder might want to make many copies of a particular plant quickly. The easiest and cheapest way to do this is to take cuttings. Shoots are cut from the parent plant, then the end of each...
We start again. This guy is speaking very fast, so we can listen carefully.
Can you spot the two clones? When we talk about clones in science, we mean organisms that are identical copies. They have the same DNA as each other. Identical twins are examples of naturally occurring clones. Both plants and animals can be cloned. Let's start with plants. A gardener, farmer, or plant breeder might want to make many copies of a particular plant quickly. The easiest and cheapest way to do this is to take cuttings. Shoots are cut from the parent plant, then the end of each shoot is dipped in hormone rooting powder and placed into a pot of soil. The hormone rooting powder will encourage the cutting to start growing roots, and soon a whole new plant will have grown which is identical to the original plant. The method works because the shoots of the plant contain stem cells, which are able to differentiate to form different cells and tissues. You can find out more about this process by watching this video. However, you might want to produce hundreds of plants from only a small piece of plant tissue. To do this, another method called tissue culture, also known as micropropagation, is used. Here are a few cells taken from the parent plant and placed onto a nutrient jelly using antiseptic technique. This means making sure no microorganisms contaminate the jelly. The cells will start to differentiate and form new plants. Animals can also be cloned, but different techniques need to be used. In animals, only embryo stem cells have the capability.
That's just a short video just to look at what they do when they are cloning plants. Now, what they mean from a tissue culture, it means, let's say this is one branch of the plant, one branch. They just take a small piece of the branch, very small piece, and they put that in the jelly, which is scientifically made to help the plant grow out from one shell. So they take that small shell, it's microscopic small, they put it inside the jelly, and within a couple of weeks, you will start seeing the plant grow. And that's how they make many plants out of only a few. That's one way we can help to stop global warming, is by planting more trees, and by cloning and planting them, we can make more and more, and that can also help us to decrease the global warming, because the plants inhale carbon dioxide, which is part of their cleaning process and part of their photosynthesis process. So yes, that's why it's always important to have plants around. Plants keep us alive. We don't always know that, but that's their main function. They have to keep us alive. Let's quickly move on. Where's my slide now? Yeah, no, yeah. Okay. So there's something to think about. It would be nice to be able to do this in class to see how the experiments will work. So maybe in the future we can do that. Know it. Where they say know it in the book is usually very important information. So let's quickly have a read through here and see what is the information that they are going to share with us. DNA. DNA is the genetic material that contains information in living things, and it is passed from parent to their offspring. So if you look at human beings, we all have our own set of DNA. My DNA and every student in this class's DNA differs. We all have different DNA types, and we all have different DNA structures. Some of us have to wear glasses because we can't see properly. That might be a defect in some of our DNA. Some people might not hear properly, or some people are born without an arm and legs. That can sometimes be through our DNA, that we are born differently to other people. But the DNA is what makes us human. Without DNA, or it makes us a living organism, is the DNA, because that builds our whole body. That tells every single cell what to do and where to do it. So if your nose is itching and you have to scratch it, it's part built into your DNA, and your brain then connects the dots. You scratch your nose, there's a signal that's coming from your brain telling you to scratch your nose. Okay. A gene is a selection of DNA. So they choose different types of DNA. Each gene controls a particular characteristic. For example, flower color. So what they would then do during like tissue culture and crossbreeding different plants, maybe you have a red rose that does not have any smell. It doesn't smell like anything. But you have a white rose which has some of the most aromatic fragrances and smells. It just smells amazing. So then they will crossbreed those genes and hopefully they get the white rose with a red color. So they get the smell of the white rose and the color of the red rose. So then the red rose will also smell amazing. So that's what they try and do with matching different genes together. That's how some of our fruits we get do not have big seeds anymore. You know, like an orange used to have a seed that about this big and quite a few of them inside. So there was not a lot to eat in an orange. But with all the crossbreeding and all the propagation of different genetics, they've made the seeds to be very small. And now there's more flesh from the plant that we, or from the fruit that we can eat. Very smart people that thought of these things. And then a chromosome is a long piece of DNA that contains many genes. Chromosomes are found in the nucleus of cells. Now we also have chromosomes. I think we have 23 pairs of chromosomes. Once you have less than 23 or more than 23, you usually have a defect. Some of them that have less than 23 will have like Down syndrome kids have less DNA, or sorry, less chromosomes, So at the bottom here on page 66, there are different ways to use biotechnology for propagating plants. Can be useful. So let's look at some of the different ways where this biotechnology and propagating of plants are quite useful. And we can produce plants that are resistant to chemicals or to insects. This helps reduce the amount of chemicals sprayed on plants, which both saves money and helps the environment. So if you can genetically make a plant that does not get infected by any disease or no insects comes to eat them, then you don't have to use pesticides. If you don't have to use pesticides, you can keep all your plants organic, which is much healthier to us humans, and obviously you save money by not having to spend all that money on pesticides and disease recovering agents for your plants. So for example, here they gave us genetically modified BT cotton plants are more resistant to insects, pests than traditional cotton plants because it's been breeded into them to be resistant to insect pests. So if you try and eat the root, like a root aphid will eat the roots, then the roots become hard on the outside, so the root aphid can't bite into the root and can't feed on it. Number two, we can grow plants that produce fruit that takes longer to ripen, so that the fruit can be transported without going rotten. For example, bananas and tomatoes. So bananas has its own acid inside it, which helps to how can I say? It helps fruit to ripen quickly. So sometimes you don't want that to be the case. So you try and breed it so that it creates less acid so that it doesn't ripen as fast and it can stay fresher longer. The same with tomatoes. Number three, we can develop plants that are resistant to diseases caused by bacteria, fungi, or viruses. So now, like we can get sick from bacteria, you know, get a bad tummy, or from eating a fungus, or we have the new coronavirus, which is around. So we can get infected by bacteria, fungi, and viruses, and so can the plants. They also get affected, just affects them differently than us. But if it's not treated, some of the infections can be deadly. And if we create plants which are resistant, then the plants are likely to grow better and also likely to have better yields because there's no insects or bacteria to stop it from growing at their full potential. Number 4, we can grow plants that produce a yield of product, for example, more fruit. So if you grow let's say an apple tree, you might have 5 apples growing every year on your tree. And you think to yourself, man, I wish I really had some more apples because I love apples. So then you go and you crossbreed the genetics to get a higher yield. And you plant your newly genetic modified plant or your seed, and your apple tree grows. And before you know it, the apple gives you 20 apples. So it became more. That's something that people also like to do because more is better. The more fruit a plant can produce, the more people it can feed, the more money it can make for you when you sell it. A lot of positive effects that happen if we have more of something. Okay, number 5. We can produce plants that grow well in difficult environments. For example, plants that are resistant to very salty conditions. For those of us that like going to the ocean, you know, at the ocean, the water is salty. The water is always very salty. And there is always plants that grow around the areas of the ocean, and those have modified themselves to be less impacted by salty conditions because they have to live there. So their genes adapt and change to be more resistant to that. The same in very cold or very hot places. Like a lot of plants that grow in Thailand will never grow in a place like Canada, where the environment is totally different. So that's why we get certain fruits in Thailand and other fruits in different countries. For example, the only place that you can buy I think gooseberries is in Europe. Europe or in Canada. It's the only place that it actually grows in. Black currants. I think it's gooseberries. Okay, that grows only in one part of the world, one specific plant. So if you want to eat every fruit in the whole world, you'll probably have to travel to most of the countries in the world to experience that. Good. Our last little bit, let's quickly read this voice bubble from this young student here. Although biotechnology has a lot of advantages for producing new and useful types of plants, we also have to think of possible negative effects that may occur. For example, one type of crop plant may be genetically modified to be resistant to herbicide. However, if that resistance were to be transferred to wheat plants, those weeds would also now be resistant to herbicide, which could be a big problem. Therefore, we should always be careful when using different methods of biotechnology. So, yeah, for example, let's say you have certain plants like weeds that grow that you don't want them. So you spray some herbicide, which is a plant-based chemical which kills certain plants. Now, if you have a plant that in your garden which is resistant to herbicides, it doesn't get affected. And that plant somehow manages to spread that gene to the weeds around it. You will not be able to kill the weeds with herbicide. you'll have to go and cut them yourself or pull them out yourself. So while biotechnology can be very good, it can also be bad. With every good thing, there can always be a bad thing. If we do not adopt the good thing properly. If we try and do a lot of different things, then a lot of bad things can happen. Like while testing in a lab, one day, a young scientist discovered AIDS. AIDS, if you don't know, HIV is a man-made virus. It was produced inside the lab. So yes, while experimenting might be good, it can also have a very negative effect. Like HIV is one of the top killers, still one of the top killers in the world, and there is no cure for it. So yes, it's always fun to do experiments, but make sure you do your experiments in a safe environment and try and do your research before you do the experiment. So a scientist is a person who is an expert in at least one area of science. Scientist applies to scientific method when carrying out their research. So yes, we learn all this information, but will we ever be able to do it? Of course. Of course, we can also do very basic forms of biotechnology, like cuttings, for example. In the video we watched, me and you, we can all take cuttings of plants and replant them. We just get a little rooting hormone, which you can buy at most plant shops. Will have a little bit of rooting hormone. It can either be a gel or a little bit of powder, just helps the roots grow faster. So all of us can also do a little bit of biotechnology if you want to. If you have a beautiful tree at home or a bush that is very beautiful and you cannot buy another one or you cannot find one, you can take a cutting and replant it, and then you will have two. So yes, we all can do our part if we want to in creating more plants and different types of plants. Okay. Oh, a lot of information. But yes, that's all that we're going to do today for science. Ooh, I feel a little bit tired of all the speaking. Can anyone tell me, what did we speak about? Who listened? Let's hear. What did we speak about today? Anyone? Oh, okay. Seems like we're all a little bit muted. That's fine. Let's play a game of rock, paper, scissors. Let's see who can beat the teacher today. Let me first stretch. Get ready. Because the teacher doesn't like losing. Stretch your hands and your fingers so that you know that you can make the right shape. So you open your fingers like a scissor, and you stretch them. You stretch your fist and the open hand. Getting nice. Who's with us today? I see it's Aydin, it's Poom, it's Posh, it's Shen, it's Leon. Who is with us? Nam Nyang is with us. Jasmine is with us. Okay. Are we ready?
Ready.
Are we steady? And John Pon's with us.
Ready.
Okay. On 4. 1, 2, 3, shoot. Oh. Oh, only one person beat me. That's Posh. Well done, Posh. Posh is the one.
Yes.
Everyone else, jump on as a draw. The rest unlucky. Sorry, you didn't make it today. The coach will go on the list. for the prize at the end of the semester. So thank you very much for the day. I hope you guys have an amazing day. And I will speak to you again tomorrow morning, nice and early for mathematics. Ooh, nice. Mathematics. Okay, have a good day and I'll see you tomorrow. Bye-bye.
Bye bye. Bye bye.
Bye bye.
Bye bye.
Bye bye.
Bye bye.